pertussis. So those are the organisms for those, and what those cause. And I'm sure most of us have had a Tdap or DTaP. And then the Hib vaccine is Haemophilus influenza that can also cause meningitis. And anthrax, we don't vaccinate for that really. I've heard that that's kind of a thing coming up. Cholera, I've heard that's kind of a thing coming up. Working tuberculosis and Clostridioides, but these ones are still, we don't use them very often because I don't know if they're not very effective. It's probably because they're not very effective. Okay. So here's the ones I want you to remember. Okay, so now identification, we're just gonna kind of skip over identification because this is what we do in the lab. So in the lab, we look at bacterial structure. So we can straight away know if it's a gram-positive or gram-negative. We can look and see what shape it is. We can look and see what arrangement it's in. We can see if there's mycolic acid on the outside is what we did last time. We did endospores, so we could see if that bacteria forms endospores. So today we're gonna look at blood exotoxins. We're gonna look at exotoxins today. We're gonna look at fermentation today. We're gonna look at halophiles today. We're gonna look at lactose fermentation today. So what we can do in the lab is basically what we're doing is, is your patient is sick. So is it a gram positive or a gram negative? Okay, well, if it's a gram positive, I gram stained it. Now, does it, what's one we've done so far? Does it form endospores? It does not form endospores. Yes, it does form endospores. So what we're doing is we're not just doing all of those labs for fun. Yes, I think it's fun. I love doing all the labs. But what we're doing is we're identifying. We're using techniques to be able to identify those bacterial cultures and what they are and how they make people sick. So that is what the whole point is identification. And we have funky ways of identification because bacteria are hard, they're difficult. Like they're not, if we could just look at one test and that one test would tell us what bacterial infection that our patient had, sweet. Well, we can't. We have to do multiple tests to try to figure out what our patient has because that's all we've come up with. That's our best techniques that we know about. Yeah?
Is there any kind of a flowchart like that we can look at?
Candy question. You're gonna make one. I'm running low on candy. We're going to make one in the lab. So in the lab yourself, you're going to make a flow chart. But what clinical lab scientists do is they have a checklist, and actually now it's all computerized, so they can go click, 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 click. It's staph or whatever it is, So, but it's an algorithm that is a flow chart. Basically, is what the computer is doing, is creating a flow chart to tell you what most likely it is that your patient has. Okay. So how do we treat bacterial infections? This is tricky. How out of cholera, what do I want to say? Tetanus. We've had every patient's wound that has gone rancid since the beginning of time, and all of the different things. I could go out on the beach and stub my toe and essentially die from that infection. I could. But we have one tool in the tool chest. Once the bacterial infection has set in, we have one tool. And that one tool is antibiotics. So this is why I'm emphasizing how crazy this tool is because we've almost done nothing, which now I can't say that that's true so much. People are trying to do things, but we've almost done nothing to protect our one tool that people have used since the beginning of time, since the 1940s, 1950s. But before that, millions and millions of people died from infection. We have this one cool tool that we figured out. Now we have spread that tool all over the world 100 million times over, and now this tool is going away. So what do we do now? We don't really one tool, if the tool is not effective anymore, we're gonna go back to the Stone Age of when we're all dying from a toe infection, because now the antibiotics won't work anymore. So we're gonna talk about resistance, how antibiotic resistance works, and why those tools are becoming less useful. Let me see, I don't think we have enough time to watch these, but I will ask you questions on these quizzes, so I want you to, on these videos, because I want you to watch them very badly. Because these videos, they're short, they're from NPR, my favorite, but they're short, and they go back from the beginning and kind of work your way up to antibiotic resistance and how that happens and how antibiotics are effective. So those are cool. So there's a couple of videos here, there's another video here, and I think that's it. Is there a third? One, no, there's two. Two videos, they're short. Okay. So the discovery of antibiotics. So now we've talked about Robert Koch. We talked about who are our first scientists. Let's do a little review. Our first scientist was Robert Hooke, who invented the first microscope, strongest first microscope really that was effective at looking at things about 300 times, and he wrote the book Micrographia. Then who was our next microbiologist? Was Van Leeuwenhoek. So Van Leeuwenhoek was improved on the microscopes, made a ton of microscopes, really successful at that, and also coined the term animalcules, probably the first protozoa, the first microorganisms that we didn't know there were organisms that were smaller than the organisms that we could see on the planet. So he was the first one to discover microorganisms. So then it was, who's our third? Pasteur. So Pasteur was the one who figured out that those little things, whatever those were, might be making us sick. What are those little things? And that actually, that's where we come from. We come from little things. And our DNA can mix together with sperm and egg and come up with a baby. Well, also that there might be tiny things in wounds or in food or everywhere, and that stuff could be making us sick. And then our last scientist that we talked about was Koch. So Koch was the one who came up with the like a scientific methodology to prove what Pasteur was saying, was the postulates to say, okay, if there's a mouse that's sick, I can culture that, which is what he discovered as well, how to culture bacteria. And then he could figure out how to culture bacteria, and then once it's cultured, he can inoculate a healthy mouse. That healthy mouse would get the same original infection. You could plate that, and both plates would match, both diseases would match. So that was really effective methodology in proving all kinds of diseases like tuberculosis and cholera and all that kind of stuff. So here's our fifth scientist is Alexander Fleming. So this is, you Google it and everyone will say the most influential scientist ever. But we're pretty glad that he's been around. I've had antibiotics before a couple of times, plus I've had surgeries, which is never a good idea if you don't have antibiotics around. Imagine having to have surgery without antibiotics, and before they even knew what aseptic techniques were, so there was doctors going from patient room to patient room, operating on the next patient, and delivering a baby with hands from a patient who had gangrene, So the chances, it was just a crapshoot. It was like flipping a coin whether you're gonna make it through surgery or not. So when this scientist in 1928 found, he was culturing bacteria, and that's what he was doing for his career. I don't know if he was officially called it microbiology yet, but he was culturing bacteria, and on his agar plate, so he was studying bacteria, and he would make a bacterial lawn, and all of a sudden on this bacterial lawn there was a missing piece, and he's like, what happened? What is that? Why is there a clearing? What's going on? Well, he took that little missing piece and cultured it. onto another plate. This is something different. Okay, well, what is that? Well, now what he could do is replicate it over and over and over and over. Now he found that every time he put some of this purple stuff on the green stuff, that it would clear it and clear it and clear it and clear it. So what was going on? They were dying. The bacteria were dying. They had no idea how to kill bacteria yet, except with crazy harsh chemicals. So they were like, okay, well, let's figure out what this is. What it was penicillium. Penicillium is a fungus, a fungi. So let me erase this. So it was a fungus growing on this plate, that it was contamination. So he didn't use proper aseptic techniques. He left his plate open too long. Penicillium is in the air. And so it was something that was in the air, you know, just like you leave your raspberries out on the counter and they grow mold on them. That's what it was. So it's a mold. It's a fungal organism that we'll learn all about penicillium and what it is and what it does. But it kills bacteria. So let's figure out how it does that. So this is penicillium, a cartoon of what it looks like, and this is what it looks like on an agar plate. We'll get to see what that looks like as it grows on an agar plate. And this was a crazy miracle drug because soldiers on the battlefield were getting a scrape and dying from a scrape, or getting a cough and dying from a cough. So they mass-produced penicillium organism, and turned it into penicillin, and that's what had saved thousands and thousands and thousands of soldiers' lives in World War II. So some say estimates over 500 million people's lives. I would say that's a vast underestimate. I mean, probably most people in this room have had antibiotics, if not all of us, and probably most people in the United States have had antibiotics at one point, and probably most people in all of Western culture societies have had antibiotics, and all over the world. So it says that antibiotics have extended the average human lifespan by 23 years. So we can live 23 years longer, thank you to Alexander Fleming. So antibiotics, just so you know, even though antibiotics sometimes, which I hate this, sometimes the term antibiotic is used to kill other organisms as well. But for the sake of this class, and to be very specific about what antibiotics are, antibiotics kill bacteria, and bacteria only. And that's actually what we need to emphasize, and as we move along, we're going to emphasize it over and over, that antibiotics don't kill viruses. They do nothing to viruses. You might as well be taking M&Ms. They don't do anything to viruses. They don't do anything to fungal organisms. They don't do anything to protozoa. You have to take an antibiotic for a bacterial infection. That's the appropriate prescription. Okay, so how antibiotics work. So antibiotics work in a bunch of different ways, but I'm just going to give you one main example. And this is kind of the classic example of how antibiotics work and why they work. So stay with me, because it can get confusing, but just stay with me for you'll see what I mean in a minute. Let me just clarify. So we're going to do the beta-lactam example. So what a beta-lactam is, so beta is this funky little bee. That's a Greek letter for bee. That's beta-lactam. And what they are, this is the example with penicillium and bacteria, what the penicillium does. So penicillium, a long time ago, let me just tell you the story really quick. So penicillium mold, this little cartoon, and bacteria have been around way longer than we have. So they both have evolved millions of years before we did. So they've both been fighting the who's gonna win battle for longer than we've been around. So bacteria can actually infect fungus, can infect a fungi. And bacteria or fungi have their own immune system. So their immune system is this beta-lactam chemical. So they create this molecule that will help them fight off a bacterial infection. So I know that we think of bacterial infections like we think about the cough and gonorrhea or chlamydia or staph infection or whatever it is, and we think about the way that bacteria affect humans, but don't forget, bacteria affect all of our plants and trees, and they can get bacterial infections just like we can. So penicillium mold, it tries to defend itself against a bacterial infection, just like our own immune system. So what it makes is, let's do penicillium in purple, just so we'll keep it consistent. This penicillium mold makes this beta-lactam ring. I'm not gonna draw it all the way out, but this is what a beta-lactam ring looks like. It's the square. So this is the chemical. Yeah?
Just to clarify, so the beta-lactams is the fungi's immune system?
Yes, it's a chemical that the penicillium mold makes for itself to defend itself against bacteria. Yeah, so it makes it for itself. So let's figure out how it does that. So what happens is, this looks recognizable, right? Here's a gram-negative bacteria, here's a gram-positive bacteria. Here's the peptidoglycan, here's the outer membrane of gram-negatives, here's the really thick peptidoglycan layer of gram-positives. So what a beta-lactam, so putting beta-lactam aside really quick, we have a bacteria, and as this bacteria is going through binary fission, it's gonna need new peptidoglycan on its outer layer, right? Because it's going through binary fission, now there's two. So it's building new bacteria, right? And when it goes through binary fission and builds new bacteria, it has to lay down new beta-lactam. Scratch that. It has to lay down new peptidoglycan. Yeah? So here's the peptidoglycan. Peptidoglycan. It has to create new peptidoglycan rings around the new bacteria that it has reproduced into. Yeah?
Does the new peptidoglycan happen before or after it splits?
So before. So what happens is in binary fission, if you remember, it starts getting bigger and starts getting bigger and starts getting bigger, and then the replication will happen, and then it will split off and create two new cells in binary fission. Does that make sense?
Yes.
Okay, so what happens is there's an enzyme that bacteria use to lay down new peptidoglycan, and that bacteria, or that enzyme is called bacterial, what is it called again? Somebody knows this. Can't remember what the name of it. Oh, bacterial transpeptidase. Bacterial transpeptidase. And so what it's doing is laying down new peptidoglycan. So this bacteria, or this bacterial enzyme, will add new peptidoglycan. That's its role, is to put peptidoglycan together to form it. So what beta-lactams do is they shut the mouth of this enzyme. And the way that they do that is because actually peptidoglycan looks a lot like a beta-lactam. Peptidoglycan shape is similar to this beta-lactam. So what it does is when this bacteria is living right here and trying to reproduce into a new bacterial cultures, and it's trying to lay down new peptidoglycan, this penicillium is making these beta-lactams, and now the enzyme can't lay down new beta-lactam, can't lay down new peptidoglycan. I'm sorry, I'm totally confusing you. Yeah.
So you're saying the beta-lactam bonds to the enzyme?
So the beta-lactam ring bonds to the enzyme. Now the enzyme cannot lay down new peptidoglycan.
Okay.
Yeah? So basically what it's doing is making this enzyme malfunction, and now there's no peptidoglycan. And now, because this enzyme doesn't work anymore and there's no peptidoglycan, now this bacteria cannot reproduce and it will die. So there's this beta-lactam ring. The beta-lactam ring is produced by the penicillium mold that's gonna so there's on the enzyme, bacterial transpeptidase, what it normally does is take the pieces of peptidoglycan and put it together. But because this beta-lactam ring is so similar to peptidoglycan, it's going to try and take that beta-lactam ring instead. And so now it's non-functional. It doesn't function anymore. So that kills the bacteria. So now what happens is the bacteria will die. So this is just one example. So what happened is on this original plate, there was a penicillium mold here. This penicillium mold is producing all of these beta-lactams, and all of these beta-lactams around here are not allowing any of these bacteria within this area to be able to reproduce, right? Because they can't lay down new peptidoglycan. Okay. So hopefully as you go through the slides, it will be a little bit more explanatory. You could just follow the little pictures and hopefully that makes sense. So here are our beta-lactam antibiotics. I'll show you, we're gonna do a whole chart and you're gonna memorize the chart. But cephalosporins, penicillins, carbapenems, and glycopeptides that have a similar structure, all of these have the beta-lactam ring in some way. That beta-lactam ring, we gathered those beta-lactam rings from different sources, from different fungi or different things that make that beta-lactam ring. Now we can put that in a pill. The patient can take the pill, and now that it has the beta-lactam ring, the bacteria can't reproduce because they can't lay down new peptidoglycan. Still with me? Okay. So other antibiotics have come from different places. So our fungal antibiotics, we've made some in the lab ourselves, not very many, right? Like, you don't have to memorize this, but just so you know. So some bacteria, other types of bacteria, some fungi, and some that we make in the lab ourselves are different types of bacteria or different types of antibiotics that we can use now. So there's not just a beta-lactam example. We've got gathered different ways of killing bacteria from different sources. And so we can give you some other examples as well. Yeah?
Do they all operate on the principle of inactivating the transpeptidase?
Great question. So here are the ways that other antibiotics can kill. So here is the cell wall. These are the ones that will break up the cell wall or the peptidoglycan. So these are the penicillins, carbapenems, cephalosporins, and glycopeptides are all going to prevent cell wall formation, prevent peptidoglycan formation. But fluoroquinolones prevent DNA replication. So remember when we went from DNA to DNA, they'll prevent DNA replication, and the way that they do that is they block topoisomerase. So they can block bacterial topoisomerase so it won't untwist anymore. Now the DNA polymerase 3 can't get in there, and so now it can't replicate.
Wait, wouldn't that mess with our own cell replication though?
Very good question, and we're going to talk about that a ton, but bacterial topoisomerase is different than human topoisomerase, but that's a fantastic question. Yeah?
Back on the chart a few slides ago, what does MOA stand for? Under the...
Oh, method of action?
Okay, thank
Yeah, yes, great question. Method of action, so these are all cell wall, they all target cell wall synthesis by blocking that bacterial transmembrane. Okay. So then folate synthesis, so we don't make our own folate, we have to get that from our diet, but bacteria make their own folate. So in folate is a vitamin, so folate synthesis is trimethoprim and sulfonamides. Protein synthesis. So what enzyme makes proteins?
Ribosomes,
Ribosomes. Why wouldn't this affect our own ribosomes?
because ours are not 70S.
Because ours are not 70S ribosomes. So the target for glycosamides, macrolides, tetracyclines, and aminoglycosides are for the 70S ribosomes. Yeah? Okay, so we can't really make these. We can't really come up with antibiotics on our own. I mean, we have come up with a few synthetic ways of killing, but always the natural way is the best way. Always coming up with examples of how other things, that's why we're so afraid to lose things like the rainforest, because there are organisms in there that kill other organisms. There's bacteria that kill other bacteria. There's bacteria that kill viruses, fungi. And studying those organisms and studying how they work are so vital to coming up with new pharmaceuticals of how we can kill organisms, because we have a hard time coming up with stuff on our own and making the enzymes to make that work. Our tools are very limited of how we can make things. Okay, so that gives you the example of what those are. Here's our list. Antibiotics can terminate protein synthesis for microbial cells like germs. Let's say that together. Antibiotics can terminate protein synthesis for microbial cells like germs. I just got this on Google. This is our mnemonic. Antibiotics can terminate protein synthesis for microbial cells like germs. Aminoglycosides, cephalosporins, tetracyclines, penicillins, sulfonamides, fluoroquinolones, macrolides, carbapenems, lincosamides, glycopeptides. That's what that mnemonic is. Here are some examples. So for the examples, what I want you to know is if it's a prefix or suffix that looks like the antibiotic, then I want you to be able to recognize that. Some, like these mycin, it's too hard to know. I'm not gonna ask you those on any great detail because it's too hard to memorize all those. I'm gonna ask you if it's a ceftriaxone, then you have to know it's a cephalosporin. If it's an ampicillin, you know it's a penicillin. If it's a sulfa, it's a sulfonamide. So that's what I want you to remember, to recognize those. Here's the flow for fluoxetine, here's the flow for fluoroquinolones. Penems are the penems. Yeah? So I won't ever ask you vancomycin unless I tell you vancomycin is a glycopeptide. What does the glycopeptide do? Yeah? Okay. So the reason here that I put some of these in red is because these are the ones you have to remember. I mean, you have to remember all of these, but these are the ones that stick out. Here's a negative, which I want you to keep that in mind. Here's a negative, aminoglycosides. Here's a positive, macrolides. Here's a positive, lincosamides. Here's a positive, glycopeptides. Everything else is what we call broad spectrum. So broad spectrum means both gram-positive and negative. Both gram-positive, negative, everything else. So if you write this little chart on the back of your quiz, when you get your quiz, then you'll know all I have to know is negative, plus, plus, plus. The rest I can fill in with broad spectrum, broad spectrum, broad spectrum. Yeah? An easier way to remember. Here's the method of action. 30S subunit of our ribosome. Cell wall, 30S subunit. Cell wall. Folate metabolism. DNA replication, that's the topoisomerase. Here's our 50S subunit. Cell wall, 50S subunit, cell wall. So this is just get a blank sheet and fill it in over and over and over. Here, I put over here, here's our beta-lactams. I don't want you to memorize what it's used to treat. So here, let me show you. I crossed that out because you can just look at that if you want to and kind of get an idea of like UTIs, maybe ear, nose, and throat stuff, sexually transmitted infections. But these these change so much, and your own, when you go through pharmacology, if you are gonna go through pharmacology, then they will have you know a couple of diagnoses that are particular to that infection, or pharmaceuticals that are particular to that infection, but they change a lot. Yeah.
So only the beta-lactam targets the cell walls, the other ones that are targeted by different molecules from either a different organism or from the same organism.
Yeah, so if I know what you're saying, so here the beta-lactams are all cell wall, cell wall, cell wall, cell wall, cell wall. Yeah.
Right,
Does that
so
make sense?
the other ones are just from a different organism, the penicillium, I guess was my question.
Yes, they're from a different organism from the penicillium, or if they're not from an organism, if they were synthetic, whatever, you don't have to memorize that, but just that these are the ones that have the beta-lactam ring that came from a fungi at some point that has the beta-lactam ring. Yeah, awesome. Okay, so antibiotics cannot be used to fight viral, fungal, protozoal infections. I'm gonna drive that home 100,000 times. Antibiotics were designed to attack cell wall, ribosomal subunits proteins, DNA replication, metabolic processes that are specific to bacteria, so they do not also affect the host. Antibiotics can also kill beneficial bacteria. That's something to know. That's a huge point. Bacteria, and just because you take an antibiotic, it's not gonna target the one bacteria infection that you have. It will kill all the beneficial bacteria as well, if it's gram-positive or gram-negative or both. So that's something really scary to think about. Antibiotics can cause side effects, including antibiotic resistance, rash, a C. diff infection, which is a super infection. I'll tell you about what that means. Sensitivity to sunlight, Achilles tendon rupture, yeast infection, liver damage, hearing loss, teeth stains, nausea, vomiting, all kinds of stuff. So we'll talk a little bit more about that. Yeah.
Never mind, I can ask it later.
You go ahead.
I was just wondering how it can cause an Achilles tendon rupture.
Oh, you know what? I don't know. It's something to do with the connective tissue in the Achilles tendon, of how it affects the connective tissue.
But
But
it
that
doesn't-
is, I have always wondered that.
It doesn't affect any other tendons?
It's, I don't know. I don't know that, but that is a really common, it's not really common, but it is a common side effect, especially for children and children athletes. So, yeah, I'll look that up. That's a good question. Okay, so here are our chapter 10 terms: superinfection, an infection secondary to antibiotic treatment. So that's our note card for today. Narrow spectrum antibiotics, mostly only gram-positive. We'll talk about why on Thursday. Broad spectrum antibiotics, both gram-positive and gram-negative. Beta-lactam antibiotics, those are the beta-lactams. Bactericidal will kill, bacteriostatic will halt the reproduction. So it might be a good idea to go through why. What is it, killing it? Or is it stopping it from reproducing? Selective toxicity kills bacteria without damaging the host tissue. So that's kind of what we're talking about, selective toxicity. So broad spectrum is the ability to treat gram-positive, gram-negatives. Generations, as they go through generations of antibiotics, they try to kill more and more gram-negatives. That's first generation, second generation. If you hear that term, that's what it means. Antibiotics are not all the way amazing at killing gram-negatives, and we'll go over why, and you could probably already have some sense of why. But the generations are, if you hear that term generation, what that means is more effective at killing gram-negatives. And then here is, so just because it's broad spectrum, it doesn't mean it kills everything. Broad spectrum, it's maybe some of the positives, some of the negatives, so a little bit of the positives, more of the negatives, a little bit of the negatives, more of the positives. You don't have to memorize that, just so you know, broad spectrum means it's covering more gram variability. Okay, so we'll talk about antibiotic resistance next time. We didn't get all the way through like I wanted to. I'm talking way too long. But we'll start with post-antibiotic era on Thursday. Okay, so let's do our note card, and we are going to the lab. types of bacteria in the lab, right? Now you kind of know a little bit of what you're looking at. This isn't the best picture, I should have done the colors correctly, but what colors were they? Let's color them appropriately to what color they should be. Do you guys remember from the lab that we did the endospore state? This Clostridioides difficile causes, can form endospores. Do you guys remember what color were the endospores?
Kind of pink.
Green. So endospores were green, because that was the malachite green, right? And then what are the cells that are not endospores called?
Vegetative.
Vegetative. So those were pink or fuchsia. No, they weren't fuchsia. They were saffronin, right? Yeah. So they were red because we use saffronin for gram seed as well. Okay. So let's say it 3 times. Clostridioides difficile.
Clostridioides difficile.
Clostridioides difficile.
Clostridioides difficile.
Last one. Clostridioides difficile.
Clostridioides difficile.
If you're gonna work in a clinical setting, you will hear about this. And the more that you hear about it and the more that you get to know about it, the more your whole clinic or hospital or everyone will be impressed, because this will come up. Okay, so they are gram positive, and they are what we call pleomorphic, or sometimes it's called polymorphic. Sucks that there's 2 names, there just is. So that means there really are, because Bacillus cereus is a bacillus, Clostridioides difficile isn't always a bacillus. Sometimes they're coccobacilli, sometimes they're bacilli. They have different shapes. They have many shapes. They're modal, they're anaerobic. They're nosocomial. What does nosocomial mean? Hospital acquired. And a lot of times they will be caused from a super infection. We just learned what that term means. Does anybody remember from 1 slide ago what that means?
They're a side effect of antibiotic use.
Fantastic, both of you. They're secondary, it's an infection from a secondary to antibiotic treatment. Super infection. They're endospore forming. Here are the toxins. I don't want you to know those. They're gray. You don't have to know what types of toxins. Enterotoxin, cytotoxin B, enterotoxin A, cytolethal distending toxin. That sounds terrible, right? Gastrointestinal diarrhea and colitis. And then pseudomembranous colitis. So pseudomembranous colitis, I'll show you a picture of what that looks like. 500,000 people per year, heck ton of people die from C. diff. 9% ends in death. If you have C. diff, you have 9% chance of mortality. That's terrifying. Reoccurring, why do you think it might be reoccurring? Endospores, awesome. You can get rid of the endospores, right? Fecal oral transmission is how it's transmitted. Let me just show you a couple of these pictures and then I'll come back for it if you need to keep writing. So this is where Clostridioides difficile is common because of the super infection, right? And because it's nosocomial. And yeah, there's all kinds of, you'll hear all kinds of stuff. Here's the pseudomembranous colitis. This is a patient's colon. This is these little pseudomembranes. They're false membranes, and they have these little pustules. And this is why the diarrhea is so foul-smelling and terrible, is because the patient's colon is no longer functioning in any sort of way. And what the colon does is it absorbs water, reabsorbs water from the feces, so if it can't reabsorb water anymore, it's water mixed with all of the leftovers. Okay, so get all cleaned up when you get down there. We're doing 3 of our pre-labs today, so maybe you can work with Dr. Rosevear to get those checked off. And then we kind of have a shorter lab, so that's why I went a little bit long, which I do every time. So when do you always start on these? I messed up on this one.
It's just such a.
I feel like I